Welcome back to Science in Real Time. I'm Carli Reyes, and our segment today is called Molecule Talk, where we break down specific papers, technologies, and ideas that are pushing biotech into the future. Today's episode is about something very fresh, a preprint posted in June 2025 called CellCLIP, Learning Perturbation Effects in Cell Painting via Text-Guided Contrastive Learning. Now, before we dive in, a disclaimer that this work is on ArXiv, which means it has not yet been peer-reviewed. The data and claims are preliminary, but the idea itself is very provocative. It tries to link images of cells with natural language descriptions of perturbations, and if it works, it could really be a step forward towards making complex cell imaging data much more interpretable and useful across disciplines. So, let's break it down. What cell painting is, what CellCLIP does differently, why it matters, and what open questions we should keep in mind as this field continues to develop. Let's start with the basics. What is cell painting? Cell painting is a high-content imaging essay. So the idea is to stain different compartments of the cell, meaning the nucleus, the mitochondria, the endoplasmic reticulum, and even the cytoskeleton, all with fluorescent dyes, all at once. You can then capture those images at high resolution and then extract hundreds or even thousands of morphological features. The end result is what's sometimes called a morphological fingerprint, a very detailed profile of how a cell looks and changes under different conditions. Why is it so powerful? Because cells respond in very subtle ways. A drug that inhibits a kinase, a CRISPR knockout, or even a toxic chemical might each leave behind distinct morphological clues. If you can profile those patterns systematically, you can then start predicting things like mechanism of action, toxicity, or even new therapeutic opportunities. The challenge really lies in interpreting those fingerprints, since it really isn't easy. You end up with tables of features like heraldic texture metric number 7, or radial distribution intensity in channel 4, so it's not really exactly intuitive. So here's where the new preprint comes in. The team behind CellCLIP asked, what if we could translate those morphological fingerprints into a space that's aligned with how biologists actually talk in language? Their approach is inspired by the CLIP model in computer vision, which stands for Contrastive Language Image Pre-training. CLIP learns to connect images and text in a shared space. That's why, if you show CLIP a picture of a dog, it can correctly rank the word dog higher than the cat or car word. CellCLIP borrows that same philosophy but for biology. So it basically works by having on one side you feeding the model cell painting images from perturbation experiments. On the other side, you feed it text, things like drug names, target pathways, or even gene knockouts. The model then is trained so that the image embeddings and the text embeddings line up in the same shared space. In theory, this means that you could ask the model, show me all the perturbations that look like kinase inhibitors. Or the reverse. Here's an image. Find me the text description that best matches it. That's incredibly powerful because it could make phenotypic screens not just something about clusters or features, but really about concepts that we can interpret and act on. So, what did the authors actually demonstrate? First, they ran retrieval tests. So for example, given an image of cells treated with a particular drug, could the model correctly pull up the right description of that drug from a pool? They report strong performance compared to other baseline approaches. Second, they evaluated mechanism of action classification. The model could better separate compounds by their mechanisms compared to traditional embeddings. And third, they looked at generalization. So, could the model handle both genetic and chemical perturbations? And the preliminary results show that it can, at least better than prior approaches. It's early days, but the concept is validated enough to spark excitement. If CellCLIP or similar approaches pan out, the implications are ginormous, starting, for example, with interpretability. Instead of staring at hundreds of abstract images of features, you could anchor cellular changes to words, so anything from drug classes, pathways, and even phenotypes. That would certainly lower the barrier for scientists to be able to use morphological profiling. We can also think about cross-modal integration. Imagine linking cell images with literature text, with omic datasets, or even with drug annotations. Suddenly, you can navigate biology across different data types in one embedding space. And last but not least, drug discovery. This could make it so much easier to discover connections. For example, recognizing that a compound might look like an immunomodulator in cell morphology, even if chemically it's still very different. Of course, there's plenty of questions that remain, starting with, can the model handle new drugs it hasn't seen before? Or how much does it really learn biology versus just memorizing labels? And how about what kind of data is more useful? Should we give it drug names? Should we prioritize target pathways? Or even structure ontologies? And how well will it scale to massive data sets we now have, like jump cell painting, for example? These are the kinds of questions peer review and replication by other labs will need to answer. So, here's the takeaway. CellCLIP is a fresh, preliminary idea that tries to bridge the gap between how cells look and how we describe biology in words. It's not peer-reviewed yet, but if the concept holds, it could really be a step towards making high-content imaging data more interpretable, searchable, and most importantly, actionable. And that is a wrap for today's episode of Molecule Talk. If today's dive into CellCLIP sparked your curiosity, you'll find the preprint linked in the show notes, along with ways to connect with us. And remember that this is just the beginning of the conversation. Every week, we'll bring you closer to the breakthroughs shaping biology in one shape or form. If you enjoyed today's episode, remember to hit subscribe, share it with a colleague, and help us grow this community of curious minds. Until next time, I am your host, Carli Reyes. Stay curious.